on this Friday night. 35 million people now under lockdown.
Now to growing concerns about the deadly coronavirus.
As the coronavirus outbreak.
The ability of the deadly coronavirus to spread is getting stronger.
In late 2019, a novel coronavirus emerged in the wet markets of Wuhan, China. The virus, which causes the disease, now known as COVID-19, presumably originated here since 27 of the first 47 people to ever be infected with it had some link to this single market. Although COVID-19 began in a single Chinese city, its presence is endured globally. After just 6 months, the virus has spread to all parts of the world, including America, who was hit especially hard. Now, the new normal is a chilling take on what life once was. Scenes of a world in a pandemic flood the news with masses of people in masks in hospital beds, crammed into uncomfortable spaces. Dystopian images of disinfecting procedures symbolize the general population's sad decline in trust of public health. Some refer to it as a war against an invisible enemy, a war in which our soldiers are the healthcare professionals working on the front lines to collect samples for testing and treat patients as best they can. Researchers behind the scenes are working just as diligently to analyze the test samples, as well as searching for possible therapeutics against this novel coronavirus. Numerous different drugs are being tested in the hopes of finding promising and reliable results. Unfortunately, the intrinsic sluggishness of reliable scientific findings does not appease world leaders who have reputations on the line. For some time now, Donald Trump has touted an anti-malarial drug, hydroxychloroquine, as the magic pill to COVID-19's complications.
Hydroxychloroquine, which I think, as you know, it's a great malaria drug. It's worked unbelievably. It's a powerful drug on malaria, and there are signs that it works on this, some very strong signs.
While it's true there have been reports to suggest hydroxychloroquine may be beneficial, the previous findings may not be as strong as Trump hopes. Here in this video, we add to the debate by discussing an academic correspondence to the editor of Cell Discovery that sheds light on the safety and efficacy of chloroquine and hydroxychloroquine, as well as explores the mechanism by which these two potential therapies interact with the novel coronavirus. In the words of the president,
And we'll see if it works.
Thank you, Chase. The paper provides 3 aspects of the drugs: the cytotoxicity, the effectivity, and the mechanisms of the drugs. The cytotoxicity, of course, as it suggests, it describes the potency of a drug, and when testing a drug, it is necessary to ask if the drug is toxic to the human body, and if it is, what other form of the drugs be safely used, which in this paper, the researcher asked which one is safer to the human body, the chloroquine or the hydrochloroquine. You don't want a drug that can make you feel depressed or sad. No, only a GPA can do that. The paper uses CC50 to describe the two drugs. CC50 is a 50% cytotoxicity concentrations of a drug. It determines the concentration of the drugs that will kill half of the cell populations. So basically, it's a survival test. The kind of cells that the experiment used is called the African green monkey kidney cells, or you can call it the Vero6 cells. And the tool that was used in this experiment is called the CCKA assay. The CCKA assay kit utilized the activity of the dehydrogenase in cells. It works by using a salt to reduce the dehydrogenase and producing a yellow dye. The amount of the yellow dye represents the amount of the cells because the two amounts are directly proportional. So by adding the different concentration of chloroquine or hydrochloroquine into a cell populations, and by using the CCKA assay kit to keep track of the cell populations, the CC50 of the two drugs can be determined. Before reviewing the finding, let's talk about what the researchers were expecting. They were expecting the hydrochloroquine will be about 40% less toxic than a chloroquine in animal, meaning that the hydrochloroquine will have a higher CC50 value because the higher cytotoxicity concentration a drug has, the safer the drug is. And that is also an ideal situation. However, the experiment reveals otherwise. It presented data that the CC50 for chloroquine is about 270 micromolarity, and for hydrochloroquine is about 240 micromolarity. The two CC50 values were very similar. In fact, by looking at the figures, the two graphs seem to coincide. But more interestingly, the CC50 for chloroquine is slightly higher than that for hydrochloroquine, suggesting that the chloroquine is the more ideal drug in terms of the cytotoxicity. Next, the paper explains about the effectivity aspect of the two drugs. Basically, it asks if both drugs are effective in treating COVID-19, which one will be more effective, the chloroquine or the hydrochloroquine. No one wants to take a drug that doesn't work, that is candy. And the big money question is that if we combine the cytotoxicity data with the effectivity data, which one would be the more ideal drug? The paper describes the effectivity of the drugs as EC50. Basically, it means have maximum effective concentration of a drug. What concentration of chloroquine or hydrochloroquine would provide half the inhibitory effect. The effective data is also done with the viral 6 cells like the last time, but in this time, RT-PCR was used for quantitative analysis instead of the CCKA assay kit. In this experiment, the cells were first pre-treated with chloroquine and hydrochloroquine compound. Then the pre-treated cells were infected with virus at different MOIs. MOIs means multiplicities of infections. It is the ratio between cells and virus. After the cells is washed and added to fresh drug medium. And lastly, the cells were quantified with reverse transcription PCR. The PCR quantification provided data that suggests at all MOI, the EC50 for chloroquine was lower than that of the hydrochloroquine, meaning chloroquine is more effective because an ideal drug would be effective at lower concentration. It also showed that the EC50 differences between chloroquine and hydrochloroquine was significant at MOI 0.01 and 0.2. This can be subject to error because we can guarantee that all cells are infected with the virus in the culture. With all this data, we can calculate the selectivity index of the two drugs. To calculate the SI of a drug, it divides the CC50 with EC50. And as noted before, an ideal drug is one that is cytotoxic at high concentration and is effective at low concentration. So an ideal drug would have a high SI value. The paper provided the SI value for chloroquine and hydrochloroquine at different MOIs, and it is found that the chloroquine has a higher SI value than the hydrochloroquine at all MOIs. And with those, we conclude the effectivity and the cytotoxicity of the chloroquine and hydrochloroquine. And I'm going to head back to you, Chase.
Thanks, Zong. After investigating the 50% cytotoxic concentration and the drug's effective concentration that gives half maximum response to determine the selectivity of the two drugs, the researchers moved to a more qualitative study to explore the results by employing immunofluorescence analysis and confocal microscopy. They did this with the intent of visualizing a detailed mechanism of action for chloroquine and hydroxychloroquine and their inhibition of virus entry. The researchers wanted to analyze the co-localization of early endosomes and endolysosomes with the virus particles in cell cultures. Previously, it had been shown that this is an integral part of infection for coronaviruses. But why is that? After all, the researchers were digging for a deeper look into how the virus incorporates itself into the cell via endocytosis. Extracellular viruses are taken up by a cell when the virus comes in contact with the plasma membrane of the cell, and the membrane pinches itself off into a vesicle called an early endosome. Here is an animation to illustrate what happens when a cell undergoes endocytosis and brings in virus particles from its surroundings. This is the early endosome that was produced from the previous pinching off of the plasma membrane and contains the viral contents within it. Early endosomes have the ability to become more acidic and mature into late endosomes. After some time, these late endosomes are covered with proteins that bind to receptors on the surface of the endosome. This targets the vesicle for eventual budding with lysosomes. Eventually, a free-floating lysosome initially meets the endosome to form a hybrid structure referred to as an endolysosome. After the endosome removes itself from this moiety, a lysosome carrying the enveloped contents is released. Ultimately, this lysosome breaks down the virus and releases its mRNA into the cytoplasm. This is where the cellular components, such as ribosomes, are able to replicate the genetic contents and produce new virus particles that leave the cell and infect its neighbors. The researchers were able to use immunofluorescence analysis and confocal microscopy to get a look at how the drugs were working in the cells. The cell's components were distinguished using a combination of dyes and fluorescently tagged antibodies. Nuclei were dyed blue. Virus particles were tagged with colored antibodies to show up as red. The researchers were also able to target certain proteins unique to endosomes and endolysosomes, which, if you recall, are the products of endosomes and lysosomes beginning to fuse. Endosomes were targeted for their content of early endosome antigen 1, EEA1, while endolysosomes were singled out for their content of late endosomal lysosomal protein, LAMP1, LAMP1. This method utilized antibodies that recognize the proteins and can be bound by another antibody with a fluorescent tag attached in order to visualize the locations of the respective organelles. The researchers first incubated the cells with chloroquine or hydroxychloroquine for an hour. Then, they exposed the cells to the virus particles for another hour. After infecting the cells, the excess virus particles were washed off. And once again, the cells were incubated with drugs for 30 minutes. The researchers then visualized the results with the previously mentioned staining and fluorescent tagging methods by taking pictures with a confocal microscope. The researchers include images to make claims from the qualitative results. The arrangement of photos is set up in a fashion that allows for easy comparison of cells left untreated and cells treated with chloroquine and hydroxychloroquine and their viral contents. As you can see, the nucleus is blue, while the free virus particles are red, and the cellular organelles under investigation are green. The top row includes pictures of when the early endosomes are stained, while the bottom row displays the endolysosomes stained in green. Included in the pictures are yellow spots where the researchers are highlighting the virus's particles that have been bound themselves to organelles. This can especially be seen in the zoomed-in portions included in the top left of each photo. One major difference the researchers noticed qualitatively was the morphology and number of the endosomes and lysosomes changed a good bit when the cells are treated with drugs. They highlight that effect by placing arrows on the enlarged organelles. Chloroquine caused the early endosomes to expand greatly without having an effect on the endolysosomes. On the other hand, hydroxychloroquine not only augmented the cell's early endosomes, it also significantly increased the number and size of the endolysosomes of the cells. The researchers counted the co-localization of the virus particles in each of the instances and visualized the results in this bar graph. What they realized was that significantly more virus particles were co-localized with early endosomes for drug-treated cells. However, a significantly smaller number of the endolysosomes were co-localized with the virus particles in drug-treated cells compared to the untreated cells. With these results, the researchers claim that the drugs were blocking the transport of the virus from the early endosomes to endolysosomes, which is a critical part in the virus's infection pathway. The quantifiable results also assert how hydroxychloroquine nearly abolished all virus particles from entering the cell's endolysosomes, while chloroquine allowed a bit more passage. The researchers surmise that this blockage is occurring due to the drug's basic properties. Previous studies have shown that chloroquine and hydroxychloroquine are weak bases that elevate the pH of acidic intracellular organelles. Not only does this cause the cell's internal organelles to swell, but it greatly hinders acidification, which is crucial for endosome maturation and function. Given the results, the researchers suggest that the drugs are inhibiting the maturation in endosomes and blocking the endocytotic pathway that the virus requires in order to infect the cells.
Alright, the time is flying fast. In the end, by combining all the data, we can sort out 3 points from the paper. The first point, from a cytotoxicity viewpoint, we know that chloroquine has higher CC50 than hydrochloroquine. The second point, from the effectivity viewpoint, we know that chloroquine has lower EC50 than hydrochloroquine. And the third point, in terms of mechanisms, we know that both chloroquine and hydrochloroquine were able to inhibit the transfer of viral material from endosome to endolysosome. So, if we compare two drugs in terms of selective index, we know that chloroquine has a higher SI value than hydrochloroquine, meaning that the chloroquine will be a more ideal drug to treat viruses. However, the paper actually advocates for using hydrochloroquine instead of chloroquine in treating viruses, even though the SI value shows that the chloroquine is the more ideal choice. But nothing is absolute. We cannot put an absolute answer in whether or not both of these drugs can be useful in treating COVID-19 or neither of these drugs are harmful to our human body, because we still lack of the research and treatments.
Things that look really good in cell culture against the virus that may look good in small animals and then don't have an impact in humans. And so those are the pieces that we're looking at.
Are really just at best suggestive.